About today's speaker, Joe Davis. He's an artist and scientist. He's the first artist to have permanent lab at the legendary MIT and also Harvard Medical School. He's also Baltan's artistic advisor, and we will work on several other future projects. Today he will inspire you with the content of his work, but also by explaining the motivations behind it. After the talk, there's an opportunity to network and have some free drinks with us, and also with DJ Lady Ida. So we hope to see you there. And for now, please welcome and enjoy Joe Davis.
Thank you. It's a pleasure to be here. I understand I was banned by Philips. That's interesting. I think that's kind of an honor. I was supposed to have used vulgar language at the Van Abbe Museum, but best of me, I can't remember using a vulgar word. I guess we'll see. So today's talk is about philosophical and ethical problems that are encountered by anyone who works with who crafts the invisible. And this goes for art and science. The title is Mnemosyne's Paradox. Mnemosyne, she was a Titan, one of the deities that preceded the Hellenic pantheon. She was thought to be responsible for the powers of reason, to give everything its designation. She was responsible for our ability to have power, to have conversation, and to call things to memory. And she had the power to give herself a name. She was the daughter of heaven and earth, and she was thought to be the inventor of all words. She was the mother of the 9 classical muses, and so the mother of all of the arts and sciences. She was responsible for history. And her name, Mnemosyne, is the word for memory in Greek. In the legends of Hades, there were 2 rivers in Hades, or 2 pools. One was her namesake, and the other was called Lethos. And also all souls drank from one or the other. So those that drank from Lethos enjoyed the bliss of total forgetfulness, and those who drank from Mnemosyne had perfect memory, absolute perfect memory. And only those souls that drank from Mnemosyne were admitted to Elysian Fields. As the inventor of all words, it's too bad she's not around right now, because some of us are having a hard time coming up with terms to describe just what it is we do. The term bioart is excuse me just a second. Yeah. Come on. Sorry. Yeah, many artists are reluctant to be classified or aligned with what the popular press has repeatedly called a movement, bioart. And the term genetic art is also sometimes used, but it would be inappropriately used for artists involved in the life sciences who use neither classical genetics nor the recombinant techniques of molecular biology. On the left is Stelarc and his famous third arm, and on the right is one of the most inspiring works of genetic art that I know, or works of art and life sciences that I know. This is Martha de Menezes' manipulated butterflies. She transplants cells from the imaginal wing discs of butterfly pupa and thus redesigns the wings of adult butterflies. But perhaps the most inspiring work in the arts and life sciences that I'm aware of, or at least one of them, is the work of this woman, Rachel Berwick. She's actually a professor of glass at the Rhode Island School of Design. She found out about Alexander von Humboldt's expeditions to the Orinoco in Brazil. This was the era of great expeditions. Captain Cook and Charles Darwin. Anyway, von Humboldt's brother, Alexander, his brother anyway, was the namesake of Humboldt University in Berlin, and he was a linguist. So when Alexander visited various cultures in the Amazon, he wrote down the rudiments of the languages he encountered to send to his brother. In one village, he encountered these parrots in the hut he was staying in that spoke a different language than the villagers. So Humboldt inquired of the villagers, well, what is this language the parrots are speaking? And the villagers went, well, that language is extinct now because we killed every last man, woman, and child a couple of weeks ago. So these parrots, these birds, were the last gateway to history, human history, that could have reached back 100,000 years. So Rachel went to the Orinoco in Brazil, found the same species of parrot, brought two of them to New York City, kept them in a language-proof room, and taught them Humboldt's language. She later used them to teach other parrots Humboldt's language. This, I think, was a great contribution to the arts. On the left is the work of Oran Katz and Yonat Zur, Symbiotica in Perth, Australia, at University of Western Australia. Guatemalan worry dolls are these little straw dolls that little kids in Guatemala take to bed, and the worry dolls are supposed to absorb all their concerns and bad dreams. And so Oran and Yonat made the substrates for tissue culture into these Guatemalan worry dolls, likeness of these Guatemalan worry dolls, and grew human nerve tissue on them. On the right are some really interesting, I think a really profound contribution to the arts that was not really carried out by artists, rather by scientists at the transgenic Silkworm Research Center at the National Center for Agrobiological Sciences in Japan, in Tsukuba. They created, 5 or 10 years ago, they created gene fusions with proteins that are secreted by the silk gland and fluorescent proteins, and the result are these really beautiful fluorescent silks. I think an important contribution to the arts. This is Hideki Suzutsu. He's the director of that facility, and I was there, spent a few months there last year doing gene fusions with a protein called fibrin, which is one of the proteins secreted by the silk gland. This is our mechanism. We used baculovirus fragments of an insect virus, to fuse fibrin, the silkworm protein, with a protein called silicatine, which we obtained from a marine jellyfish, a marine sponge. And the work of this protein is to chelate silica, glass, from seawater. It's ubiquitous in marine organisms. Some marine organisms use it to make their exoskeletons. The marine sponge Tethys that we obtained the gene from uses it to make its glass endoskeleton. But the gene is promiscuous, and so if you starve the growth media for silica and instead give it metal salts, the protein tries to sequester that metal instead. Let's see, I might be ahead of myself here. Yeah. So we use non-diapausal silkworms with only 4-month egg-to-egg cycles. Otherwise, a project that took a year and a half would have taken 5 years. But we've now obtained a stable strain, and I'm trying to get them back to the United States. We're going to feed the silkworms gold chloride in their mulberry mush. I want to spin some gold cocoons, metallic gold cocoons. I figure one in the window at Tiffany's or Pierre Cardin could fund the whole future of science and art. Some of my earlier work has been recognized as making scientific contributions. It was cited in Science this September as the first example of human intellectual information committed to the biological archive. So, the world of molecular biology, as you probably know, is anything but uniform. In most laboratories, we contain several different disciplines. In my laboratories at MIT and Harvard, we have medical doctors, biophysicists, biochemists, chemists, microbiologists, bioinformaticians, etc., etc. And these people teach each other their scientific micro-vernaculars. They work together and they publish together in the literature. So some artists in Australia and the United States have established formal relationships with the laboratory and temporary, less formal relationships with laboratories in Europe and elsewhere. But it's probably safe to say that the interdisciplinary nature of these research environments has helped enable artists to find themselves at laboratory benches. The early work of some of these artists is now increasingly recognized as having formed the basis for entirely new art forms. And along the way, they've contributed to other fields modestly, including areas like education, instrumentation, horticulture, and other fields. There's also been some hubbub about some of the more fantastic claims these artists have made and possibly alleged unethical activities. On the right of the seeds of the genetically engineered seeds of an artist, Heath Bunting in the UK. These seeds are weeds that are resistant to Monsanto herbicides. He distributes them on the web, and he's partnered with a Danish rocket company that makes rockets for political protests. This is the N-55 rocket system, launched at an angle of 85 degrees. It can carry 2 kilograms to approximately 5,000 meters, and thus distribute them over large areas of farmers' fields to protest against Monsanto. Now, I can see how they think they're protesting against Monsanto, but the true damage, of course, is done to farmers themselves who already have enough problems. And of course, there's the now very famous story of Steve Kurtz, whose apartment in Buffalo, New York, was raided in May 2004. And they found, and in all of the press, and there was a lot of it, they said these organisms found in his house were harmless organisms, but they never told you exactly what organisms they were. Well, one of them was this opportunistic pathogen called Serratia marcescens. There were also laboratory strains of E. coli and Bacillus subtilis. E. coli was antibiotic resistant. It's obviously not a good idea to arbitrarily distribute antibiotic resistance into the environment. And Serratia is generally thought to be safe for, Yes, it's safe for use in the laboratory at the lowest level of biocontainment, and even in middle schools and high schools where biosafety is part of, ostensibly part of the curriculum. But there's a darker side to this story. The United States Navy dispersed clouds of Serratia marcescens in the San Francisco harbor in 1950, San Francisco Bay, and it was detected at monitoring stations more than 30 miles away. It was to test the effectiveness of biological weapons. So if you have if you have any kind of, if you're immunocompromised in any way, if you're a transplant patient or if you have HIV, it's one of the, Serratia is one of the, known to be one of the significant causes of hospital-acquired infections, and it was implicated in the death of one man and the hospitalization of 10 others at the during these tests in 1950 at Stanford University Hospital, and these were the only ones that were reported, so we really don't know how many were actually infected. In 1977, the United States Senate severely criticized the military for these tests. It's a part of, Kurtz envisioned these as sort of mock anthrax attacks, and he used this same biological warfare simulant for testing the dispersal and spread of biological warfare agents, and he used some of these, then the purpose, of course, was to encourage dialogue and discussion among the public. And he made some of these dispersals in populated areas. At one point, he determined, became aware that Serratia indeed could be pathogenic, and he wrote his partner, Robert Farrell, who later got in significant trouble about it, and asked for an alternative. No alternative was provided, and so Kurtz decided to release Serratia anyway. He'd applied to be a customer of the American Tissue Type Cell Collection, the warehouse for organisms that science uses, a kind of library, but you have to have a laboratory with biosafety protocols in place, and anyway, because of his improvised facilities and his lack of established biosafety protocols, his right to be a customer was denied, and so he obtained these organisms illegally, and that's why he was indicted for mail fraud. On a technicality, on April 21st, 2008, the charges were dropped against Steve, and because of this grand publicity, he'd raised over $350,000 to cover the costs of his defense. Let's see. $200,000 were devoted to legal fees. $108,930 were donated to civil liberties organizations, and upwards of $41,000 are still unaccounted for. So, artists, art historians, and critics aren't only interested in life sciences because there are military applications for political reasons. The whole idea of artists working in these laboratories has also come under scrutiny, special scrutiny. I have personally been accused of aestheticizing biotechnology, of playing down perceived risks on society and the environment. There are assertions that I haven't been adequately concerned with critical reflection and discussion. Ingeborg Reicheli published this in her book, Genetic Engineering in Contemporary Art, but she's since changed her tune. She took back this indictment. But there are some real there's some real cause for concern. There are notorious aspects of life sciences, of the biotechnology, that involve corporate ownership of genomic data of rainforest species, and in some cases of human populations. There are concerns that lateral transfer, that genes for the production of some pharmaceutical or industrial material might be inadvertently transferred to unintended species. There are concerns that genetically modified species may replace wild species, and all of these are very legitimate concerns, but exactly what relationships all of this has to do with art remains unclear. A few highly questionable applications of biotechnology have gotten a lot of attention. I think even the aliens, apparently crop circles, are having a problem. But anyone really associated with the diverse products and technologies associated with the life sciences would quickly come to realize that most of this, the great preponderance of these developments, couldn't have been produced for such evil purposes. There are principles of free expression guarantee all of us the right to criticize government, industry, social norms, etc. But I don't think that's really what the problem is right now. It's more about the immediacy of these works of art, and I think that sometimes these claims to inspire debate and dialogue may actually underlie more pretensive exchanges. The facts at hand are really centered on important larger issues, but we have to admit that the significance of artistic interventions can be easily overstated. The international scientific community rightly considers falsification of data, misrepresentation of data, plagiarism, fraud, all of these things to be violations of principles of the ethical conduct of research. But an artist claiming to create something that someone else created, or claim to create something that hasn't been created at all, may still find support in the community, the culture of the arts, critics, galleries, museums. If these artistic claims are based on dubious experimental results or misrepresentations of their collaborations with respected researchers, well then we may find, as a whole, the doors to the laboratory shut in our faces. Hopelessly outdated traditions suggest that artists have no codes of ethical conduct, no responsibility to observe ethical conduct whatsoever. But if you press them, you'll find that they will admit that they don't like things like forgery and plagiarism. They do have an underlying ethical code. There were certainly times when fighting for freedom of expression was really important, as in the Cultural Revolution in China, when a huge legacy of human intellectual objects and information were intentionally destroyed. Likewise, the so-called degenerate art in the Third Reich. I had personally come up against government in trying to protect my art. The city of Gulfport, Mississippi ordered me to remove my own sculptures from my own yard, and if I didn't, they were going to throw them in the Gulf of Mexico for fish habitats and charge me for the operation. I had 14 days. Half the city wrote petitions for the work, half the city wrote petitions against the work. They never worried about what art was or wasn't before, but I took full advantage of the situation, and there were reporters, newspaper stories, and television with a 500-mile radius. The curator of the New Orleans Museum of Art came over and told everybody it was art. But see, this battle wasn't over profoundly important social issues. This was a battle over city ordinances about refuse and collections of trash on vacant lots. And it wasn't, this wasn't blowing up the Buddha in Afghanistan. So, some political art is historically significant in the history of art. I think Guernica is a good example. But most political art has a problem. You have to name an enemy, and you have to be on the winning, you have to pick a side, and you have to be on the winning side. The significance of all forms of propaganda rarely outlive the episodes of conflict that sponsor it in the first place. Creating art for purely political reasons almost guarantees its premature obsolescence, as if we haven't learned by now. My name is Ozymandias, King of Kings. Look upon my works, ye mighty, and despair. But we still depend on artists to predict the future and answer deep and profound questions about the mysteries of life. Artists are expected to interpret the world for us. We're still expected to summon the human spirit. See, there's something far more ageless and transcendent about art, something more truly universal and deeply poetic than something that crosses all political and national lines, something that is trans-political. Ironically, our ideas about freedom of expression may cause us to lose touch with the traditions of tolerance and respect for diversity that brought about the principles of freedom of expression in the first place. Some of the technology associated with life sciences and molecular biology are at the heart of hotly debated political and environmental issues. But artists who invoke artistic freedom to cross the line of ethical conduct and principles for the ethical conduct research, well, these artists may well find themselves in an interdisciplinary vacuum. Even artists who choose to carefully abide with ethical guidelines can expect certain criticism. Problems arise because most of the public in the United States and even in Western Europe are technologically illiterate, scientifically illiterate, and their common sense gut reactions leave them automatically confused. The artist says, "Here is something that's never been seen before. It's made with very pure substances that are smaller than the shortest wavelengths of visible light. It can't be resolved with the most advanced digital imaging or imaging equipment or the most advanced optical microscopes, electron microscopes. None of them can see this far. A suit of clothes made from these materials could only be detected by a very select group of people, and then only those who have special tools and special skills. As it is, even these gifted people couldn't directly gaze upon it." The artist says, "Here is some invisible art." And we say, how can this be? Seeing is believing, right? We expect if it's fraud, that if it's subject to careful scrutiny, that all fictions and fabrications will automatically unravel. The catch is that most critical observers can only suspect the truth. They can't really know it. It's art, and it's articles of faith. We take it for granted that all the pages of history are written into stone, but history is never comprehensively recorded in the first place, and memory, of course, is never lossless. The stones onto which we write our history are palimpsests, constantly overlain again and again with the marks of time and the elements and endless retrospectives. We think we've witnessed something beautiful, something wonderful, and it turns out to be nothing at all. The interval between what we see and what we remember is almost always discontinuous and completely altogether variable. Descartes said about reality that the world around us exists only because we believe in it. I can be Googled, therefore I am. And if we quit believing in it, well, the world quits to exist. And Kant took it a little further. He said, we never really perceive reality in the first place, that what we perceive is the product of the human sensorium, and the human sensorium interprets the world. A good example is optical integration time, how we're able to see motion pictures and television. The eye perceives things in a period of 0.2 of a second, And anything it sees in less than 2/10 of a second gets added up into the 2/10 of a second interval. So if I give you, like, I know from, I'm kind of an amateur astronomer, that in terms of astronomical objects, minimum visibility is 10 to the 9th photons per square centimeter per second. So if I show you 10 to the 2nd photons per square centimeter per second, you can't see it. But if I show you that eight or no, 4 or 5 times in the same 2/10 of a second, well, the eye artificially puts them all together and you see visible light where no visible light existed. So even while what we've seen still burns brightly in memory, in the conscious mind, the conspicuous and capricious nature that operates the thin apertures of belief and omission step in almost immediately to replace the truth. And where we run into problems, we only have to dust off old parables to cope with the loose ends and contradictions. We gather together truth and new truth, and we give it a name. We call it Yeti. We call it Nessie. We call it UFO. We call it Green Bunny. We give the thing a real name, and we think the thing itself is real. We're highly suggestible. Names come to the lips of amateurs and scholars, and they taste really good, and they roll around on the tongue, and we think that we think that we know something because we know the name for something. Suppose something really special happens, something that's never happened before. Looking on this new reality, we're witnesses, but we're unable to tell others what it is we've seen. Our companions are curious, determined to know what it is that we've seen, so we try to tell them. Let's suppose we peered into the first high-powered microscope in the world that's ever come into existence, and we've seen the invisible microbial world for the very first time. Our companions are curious, they want to know what we've seen, and so we try to tell them. We say where the unaided eye can see nothing at all, there are these gossamery, transparent creatures like the wings of tiny insects. There are so many of them, they're like fish in the sea. They're like, each one like a single drop of rain, like beads of glass. They're so abundant, they're like herds of buffalo on the prairie. They come in all shapes and sizes. Some are cones, some are spheres, some are cylinders, and some are like the threads of screws. Some swim like snakes, others flutter like birds. Some are sedentary, entirely sedentary, and line up in long strands like stalks of wheat. So, our friends think that creatures in the microscope are like wings of insects and drops of rain, like beads of glass and fish and snakes and buffaloes and on the prairie and threads of screws. They think that the microscopic, invisible microscopic world is like anything we tell them it's like if they haven't seen it themselves. They can't really know it, though. They can only know of it. But before long, all manner of microscopes come into the world, and pretty soon anybody who wants to can peer into the microcosm and see it for themselves. They see that while they're like wings of insects, they're not wings of tiny insects. They see that while they're abundant and like fish in the sea, that they're not fish in the sea. They see that while they're like drops of rain and beads of glass, they're not. They see that while they're like birds and snakes and fish and wheat and threads of screws, they're not. They see that while they're like all of these things, they're different from all of these things apart and together. They're like all of these things, yet they are not. Sooner or later, these people who've also seen through microscopes and peered into the microcosm, well, they run into someone else who hasn't seen the microscopic world, and so they speak of what they've seen. They say that there are tiny entities in the microcosm that are both real and invisible, but this person who hasn't seen them doesn't understand. Now, these second seers fall back and recounting the names of things that microbes are like. They know from their own experience that whatever they tell them won't really be enough. It won't really be the truth, but rather only part of the truth. They know that the person who's never seen through a microscope will not understand reality from their words, even though they have all of the words in the world at their disposal. In fact, to really know about the invisible world under the microscope, one must do much more than merely peer at them through the objective lens. Anyone who really wants to learn something about the microscopic world will want to understand how microorganisms have impacts on the environment and how to identify and culture and select them. Anyone who wants to know something about life in the microscopic world would want to understand the role these organisms play in agriculture and medicine, infection and disease. Someone seeking this knowledge would want to know how microorganisms are used to elucidate the origin of life, the molecular basis of heredity and metabolism. We'd want to know how phenotypes and genotypes are adjusted and manipulated. We'd want to know about things like cellular signaling, membrane transport, vectors, introns, exons, all of these things. We'd want to understand the central dogma about transcription and translation, about how DNA becomes RNA, becomes protein. We'd want to know about the many useful substances that can be assembled into new tools and nanoscale devices, fluorescent markers, and how entirely new factories may be built for creating clean fuels and pharmaceuticals, cleaning wastewater, and generating electricity. Whoops. More importantly, we now know that a master plan for life itself is coded into the substances found in these tiny organisms, and we want the wisdom, we want to acquire the skills and the tools we need to completely resolve it. We have to learn all of these things if we want to understand something about what there is to understand about the living, invisible world. Certainly to peer through a microscope is not nearly enough. The facts at hand are much too deeply invisible. The context of the fabric of reality is much finer and far more intricate than the kinds of things that we can see, even the tiniest objects that we can see in the compass of a microscope objective. The search for reality is the search for invisibility, and for invisibility within invisibility. Human beings tend to forget reality, however, and invent words. Whole new vocabularies are invented and new definitions are stuffed into the dictionary. Our friends think the more words we can remember, the more clever we are. But the cleverest among us cannot regard the great transformations of the world unless they've seen it in the way that it was seen for the very first time. The thing that happened before is still happening. It's still a miracle. The great mystery still squats over the world. All of the objects and images in our galleries and museums, our experiments, our findings, our observations, the wisdom and follies of our scientists and scholars, all of our works of art and science, our great beacons of learning, all of our catalogs and journals and posters and abstracts, all of these things, these are the fabric of our illusions. We think we know something, and it's nothing. It's none of the things that we've ever come up for a name for, and it's all of them. It's like wings of insects and beads of glass. It's like herds of buffalo and fish and snakes, fluttering birds, stalks of wheat and threads of screws. We are still struggling to find names for all the mysteries that trouble us. And the thing we have been searching for is still a secret. It's still invisible. To quote a 2nd century Gnostic, it is neither simplicity nor impossibility of composition, nor inconceptibility, nor imperceptibility, neither human, nor angel, nor God, neither anything at all for which we've ever given a name. Like the bones and yarrow stalks of soothsayers and prophets, science provides us with sets of formal symbols, talismans, familiars, signposts to guide us through hidden worlds. We know that notions like van der Waal radii and all of the balls and sticks and ribbons that we use to draw molecular models, we know that these are fictions. We know that all of these models are flawed, that atoms are quantum entities that pop in and out of reality, that they don't simply exist as Newtonian arrangements of planetary objects. We see the shadows of things and we think we know the thing that casts the shadow, like reading tea leaves. These are our crystal balls and our yarrow stalks, our talismans and our milagros. We now know that all of the ordinary stuff in the universe, the matter that we're familiar with, solid, liquid, gas, plasma, that this consists of only 5% of the universe. That nearly 25% of the universe is composed of something called dark matter, and that stubbornly resisted all of our efforts to observe it. And 75% consists of an even more mysterious entity called dark energy. We try to describe the universe, and we're still seers and soothsayers. But the amount of the universe, the observable universe, is so small. The universe that all human beings will ever have access to is so tiny. It's like, you can read the numbers, it's like 10 to the minus 10 to the another exponent minus 30. Good luck ever trying to write that number. But that's how much we will never see, we will never know. The great mystery exists as a metaphor. Knowledge itself is a myth and a metaphor. It's the sole temptation in the gardens of paradise, the greatest mystery of all. It's the name we've given to all the deepest secrets and impenetrable mysteries. We call it forbidden fruit. It's the name for the darkest part of our human soul. It's the name for the epic conflict, dreadful conflict of wisdom and knowledge that we've carried on for as long as Homo sapiens have walked on the earth. It's a name for all of that which has left us less than divine. So there's an apple, a wild apple in Kazakhstan called Malus cerevisiae. It grows on the slopes of a mountain range in eastern Kazakhstan that extends into China, called Tian Shan. It's a diploid apple. All domestic apples are multiploid, polyploid. It has resistance to diseases that all of the domestic apples are susceptible to. It has a larger genetic diversity, of course, than domestic apples, because we remove genetic diversity from our cultivars when we create them. So I obtained seeds and leaf tissue from several expeditions that went in the late 90s and early 2000s. It's still supposedly dangerous for an American to visit that part of the world. And I used a liquid nitrogen to extract DNA. I remember the day when I used to do my own phenolic extractions with all the chemistry, but now you get some Betty Crocker cake mix. It's D and easy. This is the Qiagen D and easy kit for plant DNA extraction. So I extracted the DNA of this ancient secret. George Church tapped me on the shoulder, who's my director at Harvard, tapped me on the shoulder one day saying, "Joe, your name came up in faculty meeting. We want to know why there's no art in the genetics department. You've got all this art all over MIT biology, but we don't have any. What do you have? Have any ideas?" So we have this atrium in the new research building that's full of 187,000 stones. I didn't count them all. I counted some square feet, layers, and then averaged in 187,000 stones, thereabouts. And the Pythagoreans, they thought, their emphasis on numbers, see, was the idea that the world actually was numbered, that all the laws of mathematics and geometry were in fact the laws of nature. And I thought I could get rid of these ugly green plastic picnic tables and mark the stones with numbers for hidden knowledge, for this metaphor for all knowledge. But the genome we expect, the genomes of the domestic apples, several of which have been sequenced, are around, small for a plant, but still around 750 megabases. So we expect the diploid to be 500 to 750 megabases. And that's way too many to put legibly on only 187,000 stones. But in the genome of Malus cerevisiae is the gene for something far more ancient, the gene for a protein that is really maybe the Eden protein. This is ribulose 1,5-bisphosphate carboxylase oxygenase. It's called Rubisco for short. It's the most abundant protein on Earth. And the reason it's so abundant is because it's very inefficient. But it's also one of the most ancient proteins on Earth. It takes carbon dioxide, inorganic carbon from atmosphere, and it turns it into food. And so when it appeared, it's part of the Calvin cycle of photosynthesis. And when it appeared on the once barren Earth, well, it brought forth all the gardens that would ever be. And so the stones will carry the legacy of this too. And as sort of first step experiment, I installed, on 10,000 stones, I installed an exhibit in a gallery in South Dakota, at the South Dakota School of Mines and Technology in Rapid City. I installed the Rubisco stones. All of them have numbers, a binary number for the DNA triplet on one side, and a decimal number on the other side to signify its place in the sequence. And the project to sequence the genome of this apple has become a huge consortium. It's really wonderful. It involves several of my colleagues from Rich Lab. It now involves Eric Lander and Broad Institute. And the Karolinska Institute in Stockholm will carry on most of the sequencing, but many apple geneticists, many scientists from the apple genetics community will sponsor will hold the genome in their archives and will carry out annotation and alignments, all these very difficult things. They're also looking for clues in evolutionary biology, and anyway, it's really exciting. And So, the DNA was actually shipped to Karolinska yesterday. This is a homemade machine. Homemade, we made it in lab. It's for stretching DNA on vinyl coated glass slides. And, this is the DNA of Malus cerevisiae. It is the oldest, most ancient mystery, and I want to learn to write out its name. You're looking a little closer at this ancient mystery than, only a few people have seen this far, than anyone ever has before. Thank you very much, ladies and gentlemen.